All right, today we're diving into what's got to be one of the biggest puzzles in all of biology, figuring out the secret shapes that make life, well, work. This is the incredible story of how scientists basically learned to see the invisible machines that are running inside all of us right now. So, let's start with a big question. You know how everyone says DNA is the blueprint for life? Man, yeah, that's true. But a blueprint is just a plan on paper, right? It doesn't actually build anything. So what are the actual machines that read those instructions and do all the heavy lifting inside our cells?
The answer? Proteins. They're the tiny, microscopic workhorses doing pretty much everything in our bodies. And here's the single most important thing you need to know. For a protein, its shape is everything, literally. That specific, folded 3D structure is what gives it a job. You mess with the shape, you mess with the job. Or you break it completely.
Okay, so if we're going to have any hope of predicting these crazy shapes, we first have to get a handle on how they're even built in the first place. It turns out proteins have 4 different levels of structure, and each one builds right on top of the last.
And it all begins right here, at the primary level. This is just the super simple linear chain of amino acids all strung together one after another. Honestly, you can just think of it like letters spelling out a really, really long word.
But that long chain? Oh, it doesn't stay straight for long. Nope. Almost immediately, different parts of it start to twist and fold into these simple, repeating shapes. You get these little coils called alpha helices and these flat, zigzaggy bits called beta sheets. This is the secondary structure, and these are like the fundamental Lego bricks of the final shape.
And the complexity just keeps building. Now all those little helices and sheets start to fold and twist and pack together into this super complex, unique three-dimensional blob. This is the tertiary structure. This is the final working shape of a single protein chain. And for some proteins, we're still not done. Sometimes multiple of these already folded chains, we call them subunits, have to come together to form an even bigger, more complex megamachine. That's the quaternary structure. So yeah, we go from a simple line of letters to these unbelievably intricate biological engines.
So you might be wondering, why go to all the trouble of predicting these shapes? Why not just, look at them? Well, the thing is, figuring them out in a lab it's just incredibly hard. It's slow, it's expensive, and it doesn't always work. And that has created a massive, massive problem for science.
And this chart, wow, it just shows you the sheer scale of that problem. Thanks to super-fast DNA sequencing, we know the primary sequence, that string of amino acids, for literally hundreds of millions of proteins. But the number of actual 3D structures we've solved in a lab, it's less than 1%. Think about that. This huge, gaping hole in our knowledge is what scientists call the sequence structure gap.
So this is the massive challenge that has kept biologists and computer scientists up at night for decades. You've got this enormous ocean of sequences and just this tiny little island of known structures. How on earth do you bridge that gap? Well, there was really only one way forward. You had to learn how to predict them.
And that takes us back to the very first pioneers who tried to crack this code. We're talking about the 1970s here. These were the first-generation methods, and they really laid the foundation for everything that came later.
You had these early attempts, like the Chow-Fasman method and the GOR method. They're actually considered the first and second generation of thinking. They were both trying to spot patterns in the data we did have, but they went about it in slightly different ways.
The first generation was pretty simple. It was all about statistics for just one amino acid at a time. It asked a question like, does glutamic acid like being in a helix? But the second generation got a little smarter. They realized that the neighbors matter. So instead of looking at one amino acid, they'd look at a whole window of them, say 17 in a row, to predict the shape of the one in the middle. It was literally the difference between trying to understand a book by looking at single letters versus looking at short phrases. So, the big question is, did it work? Well, kinda. The accuracy was hanging around 50 to 65%. I mean, that's better than just flipping a coin, sure, but it's nowhere near good enough if you actually want to, design a new drug or really understand how a disease works. The whole field was basically stuck. They needed a breakthrough, a big one. And then, in the 1990s, that breakthrough finally came. This was the third generation of methods, and they didn't just tweak the old ideas. No, they brought in a completely new, revolutionary way of thinking. And it was all thanks to one powerful idea: evolution.
Okay, check this out, because this is the brilliant insight that changed everything. The thinking was, if a protein's 3D shape is absolutely crucial for an animal to survive, then evolution is going to fight tooth and nail to preserve that shape over millions of years.
The sequence of amino acids might change a bit here and there across different species, but the overall shape, that stays the same. The structure is way more important than the exact sequence.
And this idea was put into action with a method called PhD. It was a multi-step process. First, it would dive into a massive database and find all of a protein's evolutionary cousins. Then, it lines them all up, this is called a multiple sequence alignment, to see which spots have changed and which have stayed the same over eons. This creates this incredibly rich evolutionary profile that you then feed into a neural network, which is just a super powerful computer system for finding patterns. It learns to connect that evolutionary history to the final 3D shape.
And the result? It was a massive leap forward. For the very first time, prediction methods like PhD consistently smashed through the 70% accuracy barrier, getting up over 72%. This was an absolute game changer. Suddenly, these predictions were actually reliable enough to help guide real experiments in the lab.
But of course, science didn't just stop there. The next big idea was actually pretty simple. If one really smart method is good, what if you combine the predictions from a whole bunch of different smart methods? Maybe they'd be even better together.
And that's exactly what led to what we call consensus predictors, like a famous one called JPRED. You can really think of it as a wisdom of the crowd strategy. JPRED basically takes the answers from a whole bunch of different prediction programs, each one looking at the problem from a slightly different angle, and it combines all their votes to make one final, super robust prediction. It's like getting a second, third, and fourth opinion all at once.
And by combining all these different approaches, modern predictors can now hit accuracies of up to 82%. I mean, think about that journey. From barely better than a coin flip in the early days to over 80% today. It's just a remarkable story of how science works, you know? Building one idea on top of another until you have tools that are genuinely changing modern medicine.
But of course, the quest isn't over. Not by a long shot. While we've gotten unbelievably good at predicting the shapes of a lot of proteins, some of life's little machines are, well, they're still holding on to their secrets.
Right now, scientists are taking on even tougher nuts to crack. You've got things like transmembrane proteins. These are the ones that live stuck inside the oily membrane of a cell, so they play by totally different rules. And then you have these wild things called intrinsically disordered proteins, IDPs, which are basically biological shapeshifters. They don't even have a fixed shape at all until the moment they grab onto another molecule. These are the next big frontiers.
And that really brings us to the final and maybe most important point. This whole 50-year quest to predict protein shapes, it's not just some abstract scientific game. This is what's unlocking our ability to understand diseases on a molecular level. It's how we design new drugs. It's how we can even engineer brand new proteins to tackle global challenges. So the real question we're left with is, now that we can finally start to see the secret shapes of life, what are we going to do with that power?